Welcome to episode number 9 of All About Phage Therapy. I'm Pranav, founder of Vitalis Phage Therapy, and today I'll be in discussion with Dr. Nachimuthu Ramesh. He's a phage scientist from India who wears many different hats and dons many different roles. From having published more than 60 papers in the area of antibiotic resistance and phage therapy research, to creating and patenting a process for long-term storage of phages, to conceiving and convening India's first international phage therapy conference last year in December 2019 in his university, VIT Vellore, to being a mentor and teacher to hundreds of budding young phage scientists. Dr. Ramesh has plays many critical roles in the landscape of phage research in India. Welcome to this discussion, sir. It's a pleasure to have you here.
Thank you, Mr. Pranav, who given this excellent opportunity to join this great session. Since this is the 9th episode, I'm going to join. And I very good morning and good afternoon and good evening to all the viewers around the globe.
Right. It's good evening for us in Asia. It's good afternoon for Europe. It's good morning for the US, and probably good night to East Asia right now. So welcome everyone to this discussion. We're going to be covering a lot of varied topics today because Dr. Ramesh, as I mentioned in his introduction, has many different roles that he plays. So let's get straight into the discussion. I'd like to start by talking about the recent paper that you had published in Frontiers of Microbiology, which was titled Secondary Bacterial Infections During Pulmonary Viral Disease and Phage Therapeutics as Alternatives to Antibiotics. That paper mentioned a lot of cases of secondary bacterial infections that have happened in the last 4 major viral pandemics in the last 2 decades. What is the estimated load of the secondary bacterial infections in these kind of viral pandemics, especially the one we are going through right now with COVID-19? And have the major viral pandemics, even before this one like H1N1 and SARS, have they increased the incidence of antibiotic resistance? Because during the viral pandemic, it also leads to a big increase in antibiotic consumption.
Obviously, yes, for your question. The past 2 decades, so the world has experienced 6 major outbreaks of infectious agents like the SARS-CoV in 2002, and then H1N1 influenza in 2009, then MERS-CoV in 2012, Ebola in 2013, and Zika virus 2015, the SARS-CoV-2, present. So what happens, overall, the disease and the pandemic situation. So every year the AMR issues, the antimicrobial resistance issues, it's arising. So the reason behind now using the antibiotics, the prophylactic measure for especially in this case. So the doctors prescribing, so in our paper we mentioned based on the case studies. So 73.5% for all this viral pandemic they prescribed, and only they got 24% got infected. So just see 50% of antibiotics just we are giving only for prophylactic. So if you are using these antibiotics, what happens? There is no infection, but for prophylactic they are using it. But what happens when we consume the antibiotic, the patients? So it kill the normal flora which is already present in their body, and there may be a chances of increasing the resistance. So these are the issues with this the secondary infection, and in case of the Spanish flu, so in 1819 and 1919, so because of the secondary bacterial infections, it killed 15 million people. So because of this problem, so it accounts in the current COVID. It accounts so far based on the case studies that it published. So it 15%, 15%, the secondary infection, and among 50% patients died because of this secondary infection. So obviously, this pandemic also fueled for the antimicrobial resistance.
The numbers you mentioned are really high. So you mentioned that 73% of the people who were
No,
going
no, no, no. Among the studies, 73%, okay, the clinicians prescribed antibiotics.
prescribed antibiotics, and
Yeah,
only
prescribed antibiotics.
24%
Only 24.
are suffering from
Yeah.
the secondary bacterial infection.
Yes, yes, yeah.
So definitely, this kind of increase in terms of antibiotic consumption would definitely be fueling
Obviously,
resistance.
certainly it increased. Yeah.
So the fact that in the last 2 decades we've had 4 or 5 major viral outbreaks, including the one
Yeah.
right now, would have also been contributory factors to increasing the incidence and rate of AMR, antimicrobial.
Yeah. What happens? So during this, since the prophylactic of antibiotics, so they are prescribing the broad spectrum antibiotics. The patient may infected with the gram negative or gram positive, but to be safer from their side, so they are prescribing the broad spectrum. So broad spectrum, we already lost all available antibiotics in the world. Okay, in 2009, we had a report from the carbapenem resistance. Okay, and then the 2015. The colchicine resistance and a report from China, and colchicine is a last resort antibiotic. So we lost all available, the last resort antibiotics we lost. So because of the misuse, overuse, and improper use of antibiotics. Okay, so we need a alternative that will be discussed.
So, talking of alternatives, you in your paper you advocate the use of phage-based techniques as alternatives to using antibiotics for dealing with secondary bacterial infections during a viral outbreak. What are the advantages of using a phage-based approach rather than an antibiotic approach?
Yeah. So the antibiotics, as I mentioned earlier, now it is a narrow spectrum and broad spectrum. It has two broad types. And what happens? The patients might infected with gram negative. So there, if you are prescribing the broad spectrum, what happens? It kills both gram positive and gram negative, and along with the normal flora which is present in our body. So the cost effective. So if you are starting with the first line drugs, okay, the cost is very less. So what happens when you are going with the broad antibiotics? What happens? The cost is too high when compared with the narrow spectrum. So this is the problem with the antibiotics. And then the side effect. It has a side effect. If you are using the colchicine and cyclin, it has a neuron nephrotoxic effect. So this is the problem with the antibiotics. And then if you are using phage therapy, that we focused in a phage therapy for in this article. So when they find when they see any the secondary bacterial infections during this viral infection, so we suggest to use the bacteriophages. Okay, so we can even go for a cocktail of bacteriophages. So maybe the patients may not infected with more than 1 or 2 bacterial pathogens. So in that case, if you are using a cocktail, so it will be for best. Also, it is a genus, species, and strain specific. So it will not kill other even other pathogen, other normal flora which is present in the body. Also, the cost effective is when compared with that, it's the best. So this is the important thing in using these bacteriophages to alternative to the antibiotics. Especially, we experience in this pandemic. So next pandemic, we must be aware and try to use these bacteriophages for the secondary bacterial infection.
And you mentioned the normal flora, that also plays a very critical role in the immune system. So when that gets destroyed by broad spectrum antibiotics, it actually leads to a double whammy because person is already undergoing a viral infection, and then on top their immune system further gets compromised by a broad spectrum antibiotic.
Certainly.
So what would be the obstacles or hurdles which are preventing the use of bacteriophages right now, say in the context of the current COVID-19 pandemic? What are the obstacles which are preventing this use?
Actually, since throughout the globe, the scientists are working with this bacteriophages. So everyone are struggling with the regulatory guidelines, okay, to use the medical purpose. So we must have some common and centralized regulatory guidelines throughout the globe, not only the particular country. So we must have those body. Okay, it may be from like a WHO. We must have that regulatory body. So what happens? Whatever the product now in use and the companies are using it as a product. So they are making their own regulatory guidelines, okay, for their own product. But it is not it's not applicable for other products. So when we deal with the clinical side, general common regulatory guidelines must be needed to use these pages. Also the CGMP, a good manufacturer CGMP practice, that must be needed. And also there is a lacking of the clinical trials. Okay, so clinical trials, there is no proper guidelines to do that. So these are the obstacles. Okay, and also creating awareness to the clinicians, as well as the common people also must know about this phage therapy. The people must realize, okay, they should not panic of using this bacteriophages. So this is the big challenge for the phage community, phage scientists around the globe. So these are the major obstacles for our the phage research.
Some of the obstacles you mentioned were linked to regulatory authorities or regulatory approvals not being in place. You interact a lot with regulatory bodies, especially in India. What is your experience in terms of their approach towards using phages, or at least looking at phages as an alternative to antibiotics?
Actually, no. I think you had a last session with Dr. Vinod Kumar. He mentioned that, for example, in India, if you want to use, to use the bacteriophages, a patient got infected and he's going to die, and there is no alternate antibiotics available to treat him. And in that case, as Dr. C.S. Vinod Kumar mentioned that, based on the approval from DCGI, with 2 registered clinicians in MBBS in India, they can opt for this. But even though if any urgent case, we don't have time to do right and get the permission from them. They are also busy with other things. So we need the proper regulatory approval from government of India and all, not only the Indian throughout the globe, every country except some countries already doing, like Georgia and all.
Mm-hmm. Okay. You also mentioned about the awareness, and in your position as the joint coordinator of Indian Initiative for Management of Antibiotic Resistance, you had published a paper which had described an awareness campaign that you had run on the problem of antibiotic resistance and antibiotic misuse. Can you please describe that campaign and what were the outcomes of that campaign?
Actually, the IMR, Indian Initiative for Management of Antibiotic Resistance, actually was founded by Dr. Ashok J. Tamankar. So he was the head of Baba Atomic Research Center, Department of Biomedical Sciences. So he initiated this IMR in the conference of antibiotic resistance in IIC Bangalore in the year of 2008. So at the same time, I was also looking and also planned to start an organization. Okay, so what I thought was, okay, the problem is the people, they doesn't know the difference between the other medicines with antimicrobial agents like antiviral, antifungal, antibiotics, okay, antiparasitic. So the problem, they don't know. The problem is unaware. So our target. Why don't we create awareness to the people? So what is antibiotic? What is antibiotic resistance? What do and do not of the clinician, pharmacists, and then common people? So I was looking, and luckily I attended a conference in Allahabad. There he was the keynote speaker, and he was announcing this IMR. Then immediately I met him, and we just discussed and started. I just initially joined as a local coordinator. He appointed me as a local coordinator. And when at the time I was doing my PhD at the Bharadas University, Tripoli. So I planned, okay, at present, until now, there are no program conducted, especially in AMR, okay. So I decided to conduct this program in a small, geographic location. So I decided the Tripoli, where I did my PhD. The city, it's surrounded by many schools and colleges and industries. So we fixed a date in August, and what I did was we made a brochure. Okay, that is a very important here. We made a brochure for clinicians and the pharmacists, and then common people. Okay, so the brochures are divided by 3, and all the brochures, we followed the guidelines from the WHO. Okay, what the clinicians should do, So simple example, take antibiotic as prescribed by clinician.
Hmm.
So like that, and the clinician should not prescribe without diagnosis of the samples, and the common people should not insist the clinician to give antibiotic, and they should not go to the pharmacist to get the antibiotic. So like that, we framed things, and then we converted to the local Tamil language, and we fixed the date, and then we made a flyers, okay, and we printed the printing materials about 15,000 copies of each category, and then we got a permission from our institute, and we went to city and conducted the program throughout the city, and we met all the higher officials, including top collectors too, all the medical college dean, directors, and then local colleges, schools, auto walas, bus walas, all the we met all the the peoples. The data is already available with our blogspot and also with our website. The website name saveantibioticsindia.com.
Saveantibioticsindia.com.
Saveantibioticindia.com. So there we just publishing our all AMR related activities. So what happened? That was the first program conducted from our AMR in India. So later on, it was even 2010. I did the program in a small scale. Since the funding, when I was doing the PhD at the time, we don't have the enough fund to do this. So also we are called NGO and non-profit organization. We are not getting any money from any coordinators or any organization. We are generate our own money from locally, and we are doing this program. We did the program, and also in 2010 I did. After that, after joining VIT, I just since the day I joined the IMR, my dream was I need to conduct this program through IMR throughout India. Okay, that is not an easy task. So I just taken the time from 2009 until 2016. I just made we have more than 1,500 members around India, and also we have some international members. So what I thought, why don't we nominate coordinators for all the districts and all the states? Okay. So I just contacted the persons, and during my own the conference time and workshop time, whenever I go, so I used to advertise our IMR, and we made a social media account for our IMR, and through that we got a lot of inquiry, and they want to become a member, they want to become a coordinator, they want to do the program and all. So we had excellent response throughout India. We have coordinators from all part of India. So I just discussed with my national coordinator, Dr. Damankar. So since he is the founder and national coordinator, we were discussing, and he said, yes, okay, we can do this program. So we got a permission from our VIT, and then all the persons, we made some the rule book for everyone, every coordinator, what they should do and do not, and everything. We made it and sent them, and also we made a script, 2-minute script about how to and you have to use the antibiotics, okay, with 11 different languages in India. Since we are going to conduct this program throughout India, so each and every the common Hindi is a common for everyone, and we made maximum languages. We convert the 2-minute script to the local language, and then we made a audio, and then we disseminate to all the coordinators, and then we made a brochures and local flyers and lectures and slogan competitions, road rally. We planned everything, and then every year we used to have the WHO date. The WHO used to announce the antimicrobial awareness day for every every year, November month. Okay, so we fixed that same date, the whole week, and then each and every coordinator working in a different part of India and their institution. So they will not get the exact time to do this program on the same day. So we kept for a week, okay, hold the WHO week. We also commenced, and everyone, we had nearly 41 coordinators around India. So everyone, they did the program, okay. And then we also made some script and disseminated through the community radio. Since VIT has a community radio, we use that platform too, because even that may reach only for the school and college students, but it will not reach the other peoples who are living in deep India. Okay, so we decided to use the radio, and also we contact the All India radio, but unfortunately it was a very short time. They cannot give the time. And since we have the community radio network throughout India, so we used more than 350 community radio stations. We got a permissions from the head or the chairman of this CR, and we just asked them to disseminate this information per day, morning, afternoon, and evening. Okay, so it was covered completely throughout India. It was covered. So we made a map, okay, location map and everything, and all the details are available with our published paper along with the website. Okay, they can, whoever wish to see it and whoever can wish to join it, they can join. So that was the first program in India throughout our nation. We covered with as I mentioned, the different languages, pamphlet, brochures, okay, everywhere. So that was the first initiative. And also whenever we used to meet the people, they used to say we didn't know that the difference between these antibiotics and the medicine. So that is the problem now. So they said, okay, sir, it's very useful for us. And also they said the local language, okay, what we made, that was excellent steps. So that was used for our organization, and it was a good program in AMR.
That's great. So it was all encompassing, and you said it took into account local language. It took into account all the different formats of communication through radio, through written, printed brochures.
Yes.
So, and you said that anyone can get involved with this. So that's a good information out, because a lot of people who tune into our sessions are people who are aware of phage therapy. They are aware of the power of using bacteriophages as an alternative to antibiotics and the limitations of antibiotics. So this is something that a lot of people can get involved in as an activity. The way to get involved would be to go to the website, sir. You mentioned.
Yes. Have to the saveantibioticsindia.com, and also we have a blogspot, Indian Initiative for Management of Antibiotic Resistance. There they can get all the links and everywhere. Those who are interested can send a mail, and I'll send the link to become a member. And it's a free membership. There is no cost. Life member we are offering.
Great.
Only thing there must be a biology background, and also we are now deputing some of the NGOs, because since they are interested, even though not belongs to the biology background, but they understand what is the AMR situation, what they need to do, and we are admitting them to.
Okay, that's great. What were the measurable outcomes of this program, sir? Like, was there? Did you notice a change in the consumption pattern of the antibiotics in the areas that the program was active? Did you notice more awareness amongst the people? What was the measurable outcomes that were done, that were measured after this?
As I pointed out earlier, we made a questionnaires. Okay, we made a questionnaires before and after the program. So the questionnaire is such a way that, so before that, whether they know the difference between the paracetamol and antibiotics. Okay, we made a question like this. Okay, and we circulate to we categorize into the college students, and then the medical background and pharmacology background and common people like that. And then after the program also we did the same. Okay, 1 week before we sent all the materials, hard copy materials, to all the coordinators throughout India, and somebody they received as a hard copy and made it as hard copy, and they disseminated it, okay, during before and after the program. So what happens? Luckily, surprisingly, and they ticked initially. What is the just tick the antibiotics in the list. So they ticked as a paracetamol as antibiotic. Okay, initially before, and after that program, it was exactly ticked as not an antibiotic like that. So we made such a wonderful questionnaire. So that helped a lot. And when we met the people again, the local mates, they said, okay, now we understood the difference between the medicine. And also initially we tried, if we get any cold and cough and fever, directly go to the pharmacist and get the our pill from over the counter, and we used to use. But now we are trying to maximum. Since we had a good exposure, now we are directly going to the local general practitioner, and then they're doing their steps. So it's a good thing. And we didn't do any case study after that, because scale, large scale, throughout the globe, we need throughout India, we need a lot of fun to do this case study.
So what are your thoughts on replicating this in more parts of the country? Is that something which is planned as part of the MAR program? Is it part of the agenda?
Yeah, there is organization from UK, so Antibiotic Action. So they have a funding from their local government, and they have a fund from their societies, the country societies, and every year they are also doing the same antibiotic action. Okay, from UK. So we also associated with them, and another thing, React, and there are some things they are also doing the thing. Our advice as a scientist who are now we are working in India, and we have seen the people's mentality every year, and we try to do this program all the part of India. Since our duty to create awareness to them, because of the unaware, the problem arises. The antibiotic resistance. Every day it's arising it. Every day it's arising it. We also lose everything. So we don't want to lose. Already the in-use antibiotics in future. Okay, at least we need to save this. So the program is mandatory. It's a compulsory to do every year.
Okay, right. And the interesting and encouraging part of this is that you mentioned that you could see a difference in the clinicians as well. Because clinicians are going to be the drivers of this, because they are the ones who are prescribing the antibiotics, and they are the ones who have the power to change that by not prescribing antibiotics in cases where they are not required.
Yeah.
Right. Moving from this initiative of yours, which is focused on AMR and antibiotic resistance, to a patent that you had filed on the storage process. So you filed a patent for lyophilized or freeze-dried method of long-term storage of phages. Can you share some details of that with us?
Yeah. So since the globe is moving towards the phage therapy, and it is not possible to get the bacteriophages for all the pathogenic, the PDR, pan-drug resistant pathogen, at least XDR and MDR. For example, India, I may have a bacteriophages for the top 10, the pathogen priority list of escape or like pathogens.
Right.
And some other country may not have the same phages. Okay. So what we need to exchange our phages with other countries. Okay, obviously we need to help the patient who have suffered with the pan-drug resistant pathogen. So in that case, we need to send through the sample should travel it. So the phages cannot travel with the liquid media. Okay, what happened? It may get contamination, and the problem will arise. And what we thought was, so we already whatever the methods available to do the lyophilization of phages especially. So that is not the pharmaceutically accepted and approved methods. Okay, so that cannot be the powder cannot be used as the medicines directly. What happens? That powder need to be thawed, and then it should be grown again and purified. Again they go for a injection and all. Okay, a spray. Injection, whatever. So here, what our methods differ here. We are using a different excipients. Okay, that is our main motto is to the long-term storage and stability. Stability and long-term storage is very important in bacteriophages. Okay, even if you are storing about 5 years or 10 years, it should be same. It maintain the stability, the same stability. So here we used different sugars, stabilizers. So with different concentration, and then we got at present we checked our phages, lyophilized phages until the month of 32 months. We tested it maintained the same the population, and then the stability. Okay, and now we are going to test again, and now it's running 44 months. Okay, the powder. We are just maintaining still it is in 4 degree. We kept in a 4 degree Celsius, and it's still it's maintained stability. So I can send the powder. So sending a liquid is difficult. Okay, sending powder is very easy. So throughout the globe, within 24 hours, we can it can reach anywhere. So immediately they can start the therapy. So that is how we started. And in the paper, everything available in our scientific reports paper. Whoever wish, they can start to use our methodology. It's excellent. And excuse me. And then the patent. So I cannot discuss further with that experience and the conversation what we used since already it is in a publication. So the paper is more enough for the researchers throughout the globe, they are doing the page work. I suggest this one for their work and store their pages.
Great. Thank you for sharing all of this as well. I mean, it's I know it's something which is under process. You filed the patent, so there are limitations to how much you can share. But the interesting thing is that when it reaches, you said like it's very easy to send it to different parts of the world as a powder. When it reaches,
Sorry, another important thing. The powder what we made, it is a pharmaceutically approved, okay, accepted. That can be, immediately they can, mix with the saline and they can directly inject it.
okay, that was
That is the difference between
right.
others and ours.
Okay, so when it reaches, it can be mixed with the saline and then used in the form of IV phage, or it can be used in any other way because it will become in a liquid medium.
Yeah.
Okay, that's great. And you 32 months. You mentioned that there was no change in terms of the degradation of the storage.
Yeah.
Okay, that's great. I'd like to talk to you about the antibiotic resistance and phage therapy lab that you have set up in your school in VIT Vellore. What is the work that happens in that lab? You set up that lab and now you are supervising that lab. And what is the work that happens?
Yeah, actually the lab was established with the fund of Department of Science and Technology, Science and Engineering Research Board, Government of India. So as soon as I finished my PhD, I just applied a project in a bacterial phage for MDR clinical pathogen. So I got the project with the Young Scientist Award. So I just went to VIT to start my project. So there in 2013, I just established my lab and I started to work with this phages in the year of 2008. Okay, so when I was doing my PhD, so I experienced, daily whenever you used to collect the samples, when you see the report, initially it was ESBL, extended spectrum beta-lactamase, cephalosporin resistant, third generation cephalosporin resistant. So it was keep on, it going. The year of 2008, I thought, okay, why don't we start, work with the pages again? That is why I started, and then now, after some 7-8 years, I just established this lab. And that lab, our focus is to isolate the bacteriophages, okay, against the clinical pathogen, especially gram-positive and gram-negative, especially multi-drug resistance. We focus. We in our laboratory we used to have more than 1,500 clinical pathogens. Okay, so we used to collect from diagnostic laboratory. We have MOE with them. So we used to collect the samples from them, and we used to store the bacterium, and using against the phages. So we are collecting the phages and isolate and identify against this PDR, XDR, MDR, and PDR. That is our main motto. And also we started to work with the aquaculture. Okay, so aquaculture pathogen. And also we started with endolysin. So these are the major works we have started, and we used to have we have more than 60 bacteriophages. It is already we also made a small phage bank in our laboratory. So we have more than 60 plus bacteriophages against the PDR clinical pathogen.
And what are the sources from where the bacteriophages are being isolated?
So we are isolating the bacteriophages from the wastewater, from the city, and then anywhere, any wastewater, but mostly it will be there with where the more bacteria is there, because there is a feed for the phages. So we are focusing only the sewage waters and even water sources. Even we collect from the water from Ganges. So there is no need of any approval to collect this water sample in anywhere in India. Yeah.
And in terms of the common, superbugs that get a lot of limelight, so to say, like MRSA or vancomycin-resistant enterococcus. Does your phage lab have phages which have been isolated against these?
Yes, we have phages against MRSA and vancomycin-resistant enterococci. Also, we have that because I can mention here. The superbugs, okay, pan-resistant clinical pathogen is worse than the corona. Even corona have a supportive medicines, and we are mostly 70 to 80% of the patients they are recurring it.
Yes.
And but here, when the patient got infected with PDR, if they didn't diagnose properly, and that's it. The consequences. So we focused the WHO given the list, top 10 pathogen priority list. Okay. So we are focusing those kind of pathogen, and we are also we used to isolate the phages against the Mycobacterium phlegmatis. So now we are associated with Dr. Urmi. She also joined with us, and we are also doing the same.
Right. And even in bacteria like Klebsiella, which have a very high incidence in hospital-acquired infections, that has also acquired pan resistance to a lot of antibiotics.
Yes. So we have phages for E. coli, Club, Enterobacter, and Pseudomonas, Salmonella. Right. But we categorized, we did a whole genome of E. coli, Club, and Enterobacter. So this is the first from India, especially in the whole genome sequence analysis, and it was already submitted. The data is available with the NCBA.
And the work that you're doing with phages, these are all natural phages.
Yes,
you're not working on engineered phages.
No, we are not doing any genetically modified. We are using natural bacterial phages.
Okay.
Yeah, and we are expecting that phages must have a good stability. Means not the temperature and the pH. This we were just focusing, the stable and lytic phages. Okay, it should be stable with the temperature ranging from 2 to 50. We have phages the range between 2 to 15. Also the pH range from the same 2 to 11. So it is easy to use the patients even the high temperature. It should stable during the high fever. So we kept in mind that, and whichever the phages we are working on, it should have a good stability.
Okay, all right. And other than the work of your lab, what are your other focus areas of research currently?
Research, as my focus is, I have two core. One is antimicrobial resistance and bacteriophage therapy. So we are characterizing the antibiotic resistance. Now we focus only not only the clinical side, also we are focusing the environment side. So what happens? We just getting the environment samples and finding out whether the antibiotic resistant bacterium is there or not. And we did a research from our Indian currency note whether the pathogenic bacterium is present or not. And also we did the same study with the autoteller machines, ATM machines. Okay, the keypad, whether the keypad has the things the pathogenic bacterium, especially the MDR, multi-drug resistant bacterium or not. And we did some study with the doorknob, the hospital, and then the public access vehicles. Okay, train, buses, auto, everything. So that is the environment side. And we did some poultry, culture samples from the poultry and identify the resistance. Since we are using a lot of antibiotics with the poultry, so we find a good result. And I already published everything is available in my profile. And clinical side, we are collecting the samples from the hospitals and diagnostic labs and finding out and reporting the antibiotic resistant genes. Okay, recently we also we are looking until the colistin resistance, MCR1 and 2 genes, starting with basic to high level antibiotic resistant genes. We are screening it and we are characterizing it, and then we did some of the whole genome of the MDR, and now we are analyzing the report for that things. So that's it.
Okay, that's great. You're also heavily involved in teaching, and you're currently mentoring 6 PhD students who are working in the area of antibiotic resistance and phage research. And you have mentored more than 300 students in this area.
Yeah.
Do you see an increasing interest among students to study this problem of AMR and to look at potential solutions through phage research and phage therapy for this problem?
Yes. Actually, since you mentioned past, since 2013, I'm with VIT. So every year we used to, the PG students, they need to do the project, okay, as a set project. So I used to attract, a lot of students from the, the batches, maybe from all the, category, from undergraduate, postgraduate. They wish to like to work with the AMR, and they are very much interested when they see the phages. So no, since we are covering both ages, okay, AMR and phages. So they like to join our lab. So every year minimum, we were giving a training to minimum 100 students. Okay, both semesters, from extra project, everything. It includes minimum 100 students. We are giving a training, especially in AMR and bacteriophages. Yes, so far we have given 300 plus students in AMR and bacteriophages, and somebody's got a job in the, the companies, companies, they're, antibiotics production companies, and they went for higher studies, and the BTech they went for PG, and PGs they went for higher studies. In higher studies they are doing phage work and the antibiotics field. So it attracts a lot. And the notable, the student from our lab, Dr. Prashant Manohar, he's my first PhD student. So he finished his PhD. He worked with my DSC set project, and then he joined his postdoc in a Zhejiang, university. It's an international campus from Edinburgh University. So there now he's a postdoc in the lab, and he's working with the same AMR and bacteriophages. So he's the first, and another a few of them get, they already ready. One Dr. Ms. Santini, she did her PhD in colistin resistant, colistin tricycline resistant, since it is a last resort antibiotic. And few other students, Achana and Sid, doing isolating bacteriophages in for MRSA. She's focusing mainly on GPC. And other students, MTech by research, Mr. Madhav, he did work with the aquaculture pathogenic bacterium. He isolated the phages against those. And Dr. Inian, he's doing his postdoc in my lab. So he did he's focusing the phages against the Mycobacterium sphygmatis, and also he's working with some structural proteins, and also he's focusing on the endolysins. Right. So these are the major area, and the students who already did, and they're doing good now.
Great. And what do you see the kind of work and research and career prospects for people who are right now looking at getting into the area of phage research and phage study? What do you see as the prospects for them 5 and 10 years from now, in terms of how much they'll be able to commercially apply their research in whether in educational institute or research institute setting, or even a commercial corporate setting?
Yeah, actually here I look to start with our society. So in India, we have a society for bacteriophage research and therapy. So it was initiated and by the senior professors who is working in India. She's a leader. Dr. Sanjay Shipper is the president of the society, and then Dr. C.S. Vinod Kumar is a vice president, and then Dr. Gopalnath is a secretary of this society. And then we have a lot of advisory team. Dr. Urmi, and then Dr. Taruna, and Dr. Sangeeta. So there are many who is joined hand and started this organization. And we had a great conference. Actually, first conference was happened in BHU by Dr. Gopalnath. He organized the conference. So the second conference and also first international conference in the first annual meet of our society happened our VIT. So there we just covered throughout India, and we just linked all the phage researchers in India, and we had excellent response throughout India. Also, we had 7 international speakers. All are well reputed, well known phage scientists in India. Notably Betty, Elizabeth Betty, Cutter, she joined, and there are a lot of Talman from AMR background, and Sebastian and Belinda from the China, and there are Hatful and Jeremy Bar, they could not join since they had another the commitment. So we just linked we linked all the phage scientists in India, and our motto is to create awareness to the clinicians. The clinician must know this. Whenever I used to go to any hospital, I'm going for my personal or whichever, I used to mention this phage therapy. So when I say the word phage therapy for alternate to PDR, they excellent, no, amazing, oh, good, happy news like that. They're happy that, but only thing we need to reach them. We need to create awareness to them. So our society take a lead in India. So we already doing a great job, and we are planning to conduct a annual meet and then a workshop, and we are going to start a journal, and we are going to release a newsletter of our society. Because of this pandemic, some issues, we would have released the pamphlet, sorry, the newsletter, but it was getting delaying. So we just already, fuel and going already, link the people and planning, and the motto, our job, create awareness to the people in India, the pharmacist background, the clinician background, all the those who are wish to do this. And another thing, since we lost all available antibiotics in the world, we don't have any alternate options. Okay, so we need to save the patients, those who infected with PDR. The prime, we need to go back to the phages. Okay, no other way. Since already we established, everyone has started to working, and obviously we are going to get the phages for all the the PDR clinical pathogens throughout the globe. So we cannot restrict and limit ourselves with a particular country or location. So we need to focus only the global part of scenario. So our society will play a great role, and also all the space scientists in India, and they have the great responsibilities to establish the phage in future research. And I hope we are happy about the youngsters now. They wish to start their work with phages, want to work with AMR. And also it will be good, and my future prospect about 5 to 7 years, I think we are expecting once the the regulatory body, once it's framed, once we get the good the guidelines, it can immediately we can go for clinical trials. We can, and even in India we have enough pages, more than even single. I am having more than 60 pages. So like that, everyone has, all our seniors, Vinod Kumar, Sanjay and Gopal, Taruna, Umi, and this is some notable now Sangeeta and notable scientists. They each and everyone has their has their own pages. Okay, even we started to work with Helicobacter. So we we supposed to start this, but because of this problem, we could not conduct the experiment. So like I can give a guarantee that another 5 to 7 years in India there will be a lot of product arise from our phage scientists, and also I hope the government of India will support us to do this, and also make a team for phage scientists in India. So that is very much needed. Okay, the pharma team based on the profile, and they start to deal with other countries and make guidelines, and then approve the things to the human use. So another 5 to 7 years, there'll be a great boost up in the phage research. There is no doubt in that.
That's a very encouraging note to put out for the young scientists who are looking at phage as an area of specialization. It's a positive note on which I'll start taking some questions now from people who are viewing the session, from the audience. We're getting quite a lot of questions. So let me start taking some of those questions now. There is a question from Satish Gupte, who's asking, with continuous use of phage therapy for years together, what are the long-term challenges that we are likely to come across?
So there is no limit for phages. Sometimes the phage, the bacterium become resistant and get muted to the phages. But we are very lucky enough to again isolate the phages from this, the sewage. So even we can stop the use of antibiotics, and we can keep on using the phages. Okay, what happens instead of using the antibiotics, we can now reduce the side effect, and there will not be much cost, and especially will not kill any normal flora. That is very important. The gut flora is very important for the to functioning of the body. So not only the throat, our whole body consists of weight. It is about 2 kg of flora we have. So everything is doing their job properly. So once we start to misuse the and improper use of this kind of antibiotics, what happens? We are going to disturb the normal flora, and also they will be getting a side effects. Okay, so using the phages will be the best choice. No side effect. So, since it is a genus and species strain specific, it won't kill any others. So it is very safe to use even until the end of your life.
And also the fact that if there is a phage library and there is resistance which starts building up to the existing phages which are there, new phages can always be found. It's a continuous process of isolating and finding new phages to overcome the challenge of resistance.
Yeah. So what happens, as I mentioned earlier, there may be the chances of getting mutations of the particular bacteria. But what happens, for example, to treat a person, so we'll not go with more than 10 to 15, the therapy or injection. The cycle, it may mutate 10 to the power of 50 or 150. So before that, we'll finish our therapy. So it won't mutate. Even it mutate, we'll get back from the natural. But since we have a lot of different variety of phages available, so we can use different the family can be used to kill the pathogens.
There's a question from Ajay Kumar Prajapati who's asking, can we design phages to tackle antibiotic resistance? I think maybe about synthetic phages. Can we design phages to tackle antibiotic resistance?
Yes, we can design, and we need to get the clearance from the biosafety the committee, and it can be done. It's not we can't say that no. Yes, we can do.
There's a question from Atif Khan who's asking, how do you isolate phages against bacterial pathogens or bacterial hosts which cannot be cultured in a lab?
Since we didn't work in this area, since it is not culturable, it is not possible to isolate the phages against that. Okay, still there is no methods available to do that. Maybe somebody may do, but it may not reach the scientific community. We need it must be cultured, and then we can go for isolation.
There is a question from Emily who's asking, what are the advantages of using natural phages versus synthetic phages for therapeutic purpose?
So here simple thing, when it comes from the synthetic, and they may invest a lot of money to the phages. Okay, it may cost. And also we don't know the exact for example, if a company start to produce a synthetic bacteriophages, that is their own product. Okay, that is their own product and patent. So we will not we can't see the entire genome. Okay, so what kind of things it is there, but it may be maybe some side effect may be there, but natural. So we will not see any kind of things, since we already identified and characterized. We know this complete details. So obviously, I prefer the natural phages.
There's another question from Atif Khan who's asking, what will be the shelf life of bacteriophage suspension if they are stored at 4 degrees Celsius and minus 20 degrees Celsius?
Sorry, 1 second.
What will be the shelf life of bacteriophage suspension if it is stored at 4 degrees Celsius and minus 20 degrees Celsius?
Yeah, so we did some study regarding our life-lived phages. So when we stored in a 4 degree, if you see the stability after even after 20 months, it gives a good stability and a good quantity. And when you store at 37 degrees Celsius, within a 10 months, it loses its stability. Okay, it started losing its stability. And when you want to store a phages, it's very simple to store in a refrigerator, 4 degrees Celsius. That is the best method. If you want to store in a minus 20, be cautious of using the excipients which are going to use for your the purpose.
Okay. There's another question from Satish Gupte who's asking that what is the possibility that you will be working with synthetic phages? Since you mentioned you work primarily natural.
Yeah, built in, yeah.
So he's asking what is the possibility that you will be making synthetic phages as well?
So far, no idea about synthetic phages, since we focus only the natural bacterial phage.
Right. There's a comment from Hari Sridharan who's saying, "Dear Dr. Ramesh, your knowledge and depth in the field of phages is an asset to the institution of VIT Vellore and the country. We wish you all the very best in all your endeavors."
Thank you so much. Thanks for your word.
He's also sent a question. He's asking, do you see resistance from pharma companies currently producing antibiotics against phage therapy as it would disturb their commercial viability?
See, there are no new antibiotics were discovered, and whatever the antibiotics, the last resort is the cholesterol tricycline, and there are some combination antibiotics there. Okay, so it sometimes works as a miracle. For example, if you are using individually, okay, it won't work, and if you mix with combination of other antibiotics, okay, it may like what happens with the corona. Okay, so I didn't know that there is a new drug they are using it, and obviously, as the Alexander Fleming, he mentioned that whenever you introduce any antibiotic within 2 years or 5 years, definitely it get resistant. Okay, so there is a great chances of any antimicrobial company you produce or synthesis, okay, identify from natural sources. Obviously, it will resistant within 2 or 5 years, depends on the usage.
There is a related question from Raghav Sood. He's first saying, amazing work you're doing here, Dr. Ramesh. His question is, do you have government lobbying bodies for phage therapy in India? So are there bodies which are lobbying?
No comment.
I think maybe some of the organizations like SBRT will play an important role in bringing this treatment option out towards government bodies and regulatory bodies.
There are a lot of NGOs, okay, not NGOs, and some interesting bodies. At present, we know only the Vitalis doing it, and we didn't know any other group doing this kind of things. Okay, since our society, we are focusing and spending time for the Indian researchers, and we need to our prime duty is to help them to get the method. Still, the many of them are difficulty. They are facing a problem with identifying the bacteriophages. Okay, they cannot identify it and cannot characterize it, cannot purify it. That is a problem for them. So, based on our own experience, we are trying to conducting a workshop. We are planning for the workshop and some conferences. Okay. So from our side, this is the things happening, and we don't know. Actually, I don't know who is doing in India in this thing.
There's another question from Atif Khan who's asking, are you providing your bacteriophages which are there in your phage library for the purpose of therapeutics for phage therapy for treatment?
So whatever we are characterized so far in our laboratory, obviously, if a patient get infected and he or she need to save his life, so we are ready to give the phages with some conditions, terms and conditions.
Okay. And have you had cases like that where?
Yes, I had 3 cases. Okay, from Pune and nearby, I guess West Bengal, and then New Delhi. I had a call and a mail from them, and they told that, sir, we have seen in a social media that you are doing a phage therapy and you are having a paper, and we have seen it, and there is a patient. He's the antibiotics are not responding. So can you help us like that? Actually, we are planning to I just informed her, the procedure to do this everything, and they said first we just check whether your phage is working against the pathogen or not. I said, okay, I'll send first you send your pathogen to our laboratory. We check it. Once it works, definitely I'll go for a next step like that. And unfortunately, what happens? The plant she plant, and she don't know how to send the isolate, since she's a clinical background, and she don't know how to culture it, and they may had some another problem, and it took nearly 7 days. Okay, and for to me it reached on the days of ninth days. What happens before reached to my lab, the patient died.
Okay, that's unfortunate.
So yeah, the same happened. No? Rest they called me and they given I have given the same, and they further didn't contact me. So this is the three case happened to my personal experience.
And what were the pathogens? What were the bacterial pathogens in the case?
Klebsiella and E. coli.
Klebsiella and E. coli.
Yeah.
And were these all hospital-acquired infections?
Hospital-acquired infection, and it is resistant to all available antibiotics, including colistin and tricycline. And they sent an antibiogram report. First, I asked them to send the report, and they sent it. I have seen it, and then only I asked them to send the pathogen to my laboratory. So unluckily, he died.
There's a related question, so I'll take that up right now. Are hospitals recognizing the rise of antibiotic resistance and getting interested in collaborating with your antibiotic resistance and phage therapy laboratory?
Yes. Now the clinicians, got the awareness of the MDR, and wherever I met the doctors, they used to say that, so Ramesh, it's not working, and we are interested to your phage therapy. And many cases, we used to lose the patients. Okay, since initially we don't know this thing. So when we met, when we discussed, when they go through our paper and everything, and they said, okay, so happy news that there is some alternatives there. So if you need further in future regarding this the pages, we'll contact you. And also they used to those who interested, they already they keep in touch with me, and they are updating us regarding the therapy, what they are doing it, what kind of problem they are getting it with the MDR and PDR. So now it's fine. After this, within these 2 or 3 years, I'm seeing a lot of difference with whomever I met. Okay, it may be some other doctors may be interested, and they may not reach us, and they may be only with our papers. But so at present, so far I'm contacting a lot of clinicians, a lot of doctors around India. They're saying that, so they are looking forward. They are looking for the phage therapy, obviously.
Okay, that's good. And in terms of the awareness of phage therapy, there's a related question, so I'll take that up. The question is from Meera. She's asking, do you spread awareness about phage therapy when you do the AMR awareness drives as part of your IIMAR campaigns?
So now we start now. It happened 2017. So just 1.5 years before only we started this society, and we link these people. And wherever I go, whichever the conference, yearly 6 to 7 conference I used to go to give a talk, and maybe internationally and nationally. So wherever I go, I used to represent this phage therapy and the society and the people who are there doing it. I'm just sharing the slides related to our conference and our society people and society page, everything. So I'm just doing it. I'm just started already. I'm doing it.
There's a question from Sunita who's asking about the possibility of your lab developing phages for intracellular bacteria like Mycobacterium or Mycoplasma, and also bacteria like Helicobacter pylori.
Yeah, as I pointed out earlier, I already started with working with the Helicobacter pylori. I forget her name. She's from Kasturba Medical College, and we already contacted, and they attended our conference, our wait at the conference. So she got a training, and we about to start this program. Start the work. Unfortunately, because of this the pandemic, it not didn't start. So we'll start it. But regarding the Mycoplasma and Mycobacterium, we don't have the facility. We need a BSL-3 facility. In my lab, I have a BSL-2 facility. So I'm just restricting, based on the rules and regulation, I'm just following it. So if I have I'm looking for a fund. I'm applying a fund to the government of India agencies, funding agencies. If I get it, working with broad area, no, so in gram negative alone there are a lot of pathogens are there, and also we just widen our thing with gram positive, and along with aquaculture also we started. Okay, so and we start and the endo license we start. So there are a lot of work going on, and if you are keep on expanding it, it's not possible to get the proper results. So therefore we are restricting, and maybe because of if I get enough fund in future, I may start with work with my tuberculosis. Already we contacted with the tuberculosis research center in Chennai. We have a MOU between VIT and the tuberculosis research center. The director, we already made he already visited our conference. He's the chief guest of our conference. So we had a word with them, and also we are trying with another R&Ds in Chennai. So we are already trying it. I am doing as much as I can in all the pathogens.
There's a question from Bhushan who's asking, do you prefer working with whole natural phages or license? And if so, why?
Sorry, one second.
In terms of your own research, he's asking, because you mentioned you work with phages as well as with license, endolicense. So he's asking,
Endolicense, yeah, okay.
with phages or with endolicense? And if you do prefer, then why do you prefer working with one over the other?
I'm just started with both endolicense along with the phages. Just now, endolicense just 8 months before we started. So we got a promising result. But I will write both the hearts. I will not write only one. I will write both.
Okay. All right. I think we have covered most of the questions which have come till now. If there are more questions which come up, we'll forward. Okay, there's one more question. This is from Raghav Sood again. He's asking, have you considered crowdfunding from patients who are suffering, where this has been successful, where phage therapy has been successful? Have you considered crowdfunding as a way to raise funds for further research and application of phage therapy?
Nowadays, the crowdfunding is getting famous because of some of the movies they are trying to do it. But I didn't hear this crowdfunding for research aspect. I'm here the crowdfunding, but I didn't know that, and I didn't see any kind of organization used crowdfunding for research purpose. Maybe some other countries may do, but India didn't know that. Based on my experience, I didn't know that. If there is a chances, we can do it.
I have actually seen an interesting case of someone crowdsourcing their own treatment cost to the tune of 40 to 50% of the total treatment cost by putting out their entire case history and using a crowdfunding platform to raise some money for his own treatment.
Okay, good.
So I think that covers a lot of the questions which have come in from the audience. Thank you for your patience, Dr. Ramesh, for addressing all of those questions. They were diverse, just the way your work is very diverse. The questions were reflecting that diversity, and they were all of different topics that you are working on. And even in the discussion, we've covered things from your phage research to the AMR initiatives to the conference that you had convened. So thank you so much for sharing all of this information with us, and thank you for coming on this platform.
Thank you so much, Mr. Pranav, and also I thank your wife Apurva, and for giving us a great platform to discuss about AMR and phage therapy issues. So it drives the other peoples and the clinicians and the pharmacists and the common peoples. They know that something is coming up in a future for alternative to this antibiotics. So I also hope that the countries throughout the globe they join hands and start regulatory authorities and some the pharma guidelines and to use these pages in future. So with this, I thank you once again for this episode. I thank all the viewers throughout the globe who listened to our sessions. peacefully. And once again, I thank you all. Thank you.
And actually, that's a good point to finish this on, sir. That thank you to all the audience who always, as always, tunes in and sending in your questions and comments. That really adds to the entire interaction. And it's always good to use this platform where phage scientists and phage researchers from around the world are coming and being available for a Q&A, open-ended Q&A. So we encourage you to use this platform to put your questions. It can potentially help with your research. It can potentially solve some issues that you might be facing in your work. So please go ahead and use this platform for putting these questions. All of our sessions are archived on our Facebook page. So for people who might not have been able to watch it live, you can always tune into it on our Facebook page. They're archived there. Thank you for joining in and sending in your questions and comments. And thank you, Dr. Ramesh, for
Thank you.
taking
Thank you. Also, I wish to acknowledge my institutions. So I thank my VIT management and my dean, Dr. Prakasham, who is always encouraging and supporting. So without their support and encouragement, we can't do the things. So they are supporting us to run this lab and supporting me through the seed fund. So I thank them. The whole VIT management, and then my dean and my fellow colleagues, all my research scholars, my postdoc, my PhD students, and all my trainees. Thank you all. Thank you so much.
And I look forward to seeing you again soon. Because the pandemic, everything has been shut down in terms of travel. But hopefully, when you visit New Delhi next, or maybe when we get a chance to visit your beautiful campus again, we look forward to.
We hearty welcome to you once again.
Right. All right. Okay. Thank you.
Thank you. Bye.
Bye.